Hello, again we are here with microbial anatomy and physiology. As you remember in our previous class, we were talking about the DNA replication. DNA is the basic and basic unit of the DNA bacterial cells or living organism, which is responsible to carry the information from parents to their offsprings and to have to maintain the all characteristics and all features of the living organism. So when everything is going to be divided in bacterial cell, it is also very necessary that DNA also should be replicated. So we have discussed different models of the DNA replication or DNA duplication, and the semi-conservative model we have discussed in detail. Conserved model we have discussed in detail, and Kornberg's experiment is also we have discussed in detail, which elaborated this thing in detail that DNA replication model is semi-conservative and basic units for the DNA replication are required are DNTPs, DNA template, and DNA polymerase, and magnesium ion to facilitate this reaction, which we are doing mostly in PCR every time. So if you remember in our previous class, we were talking the DNA elongation, how DNA, because we understood this thing clearly that DNA of parent cell is providing templates. By using these templates, a daughter DNA is going to synthesize. It have a specific mechanism. Here, if you will see, this is the direction of the parent DNA 3' to 5'. It is the direction. This is template or parent DNA having nucleotides adenine, cytosine, adenine, thymine, and cytosine. And these nucleotides are bonded, are together, are linked through phosphodiester bond from 3' to 5' direction. What happens when this parent strain is used as daughter strain? That thymine will bind with adenine, guanine will bind with cytosine, thymine will bind with adenine, and here is thymine, so adenine will bind here, so cytosine is here, guanine will bind with this, and this chain will increase. This chain will increase its length. And how it will form by phosphodiester bond, that it grows from 5' to 3' because it is antiparallel to parental DNA, because it is antiparallel to template. So it grows from 5' to 3'. So 3' will provide empty site to make a bond with with 5' of the coming nucleotide. So water molecule will release and here will form phosphodiester bond, and this uh, phosphodiester bond as a backbone will increase. For this reaction we know that energy is required. If you will see here, this thing which we want to explain, suppose guanine have uh, 3 hydroxyl 3' hydroxyl group is free, okay, and if the coming nucleotide is T, it is 5' site will bind with this hydroxyl group. Here we can see this molecule will release, and this 3' and 5' will make a phosphodiester bond. Water molecule will be released, and the energy which is in form of phosphate will be used for to catalyze this reaction. Again, this hydroxyl group of 3' is free, and a coming nucleotide will bind through 5' over here, and this link will gradually increase. So this process is called elongation. If we will see here, DNA elongation, this is template strain, and this is newly growing strain. If you want to see the mechanism, this is adenine. It starts from 3'. This is cytosine. This is adenine. This is thymine. This is cytosine. And this is phosphodiester bond. This is phosphodiester bond. This is phosphodiester bond. This one is phosphodiester bond. And here it is also phosphodiester bond, and it is going on. So this is new strand, or we can say that daughter strand. This is thymine, pentose sugar, and it is 3' is empty. Here guanine will bind. The 3', the 5' of guanine will bind with this 3', which was free, and it is 5' will make a bond through this, and that bond will call phosphodiester bond. Again, this is coming nucleotide. This is thymine. This have phosphate molecule, phosphate molecule, phosphate molecule. Incoming deoxyribonucleoside triphosphate. So what will be? This phosphate, which is attached in 5' of this nucleoside, it will bind with this, and phosphate molecules will be used for this reaction, to catalyze this reaction. And again here 3' is free. Okay, the coming molecule, suppose it is thymine, adenine will come over here, and the 5' of adenine will bind with it and will form bond. So gradually this chain will increase. Gradually this chain will synthesize, this chain will form. So double it is parental strand, and here daughter strand will form. Similarly, if you will see over here, this is parent strand and this is daughter strand is going to form. It is 3 hydroxyl group is free, where 5' of the coming nucleoside will make a phosphodiester bond, and with and it will increase along mechanism of DNA elongation. So in prokaryotes, there are 3 main types of DNA polymerases. So here we have to discuss, because this all process is very fast. If you will see in our preview, if you remember that 800 nucleotides are binding at per second. So speed is too fast. So this all mechanism and this all process is observed and monitored by enzymes. These enzymes are responsible which will carry this reaction and this elongation process of the DNA daughter strand. So these are enzymes. In prokaryotes, there are 3 main types of DNA polymerases which are responsible for the replication of DNA. And the replication of DNA consists of 3 stages: initiation, elongation, and termination. And these all 3 steps and the process are monitored and processed by DNA polymerase. So if you will see here, polymerase copy polymerase 1, polymerase 2, and polymerase 3. Polymerization 5 prime to 3 prime. Okay, these are carrying function polymerization from 5 prime to 3 prime direction. Exonucleases 3 prime to 5 prime, they are working, and exonucleases 5 prime to 3 prime, that polymerase is working, 1 is working, 2 is not working, and 3rd is not working. Exonuclease 5 prime to 5 prime 400, and it is not now, and this one is 10 to 20 nucleotides. 3 to 5 prime exonuclease activity, ability to remove nucleotides from the 3 prime end of the chain. These exonucleases have important proofreading ability. Without proofreading, error rate, mutation rate is 1 into 10 is the power minus 6. With proofreading, error rate is 1 into 10 is the power 9, 100-fold decreases. So 5 to 3 prime exonuclease activity function in DNA replication and repair. So this this slide tells about this. The DNA replication is controlled by our DNA polymerases, exonucleases, polymerases, exonucleases, and exonucleases 5 prime to 3 prime as well as 3 prime to 5 prime. These both are working to minimize the mutation rate, to minimize the errors, because speed is too fast, that is 800 nucleotides per second. If these enzymes are monitoring all the process, if there is any mismatch, these exonucleases will remove. How mismatch can be this? Suppose this is cytosine. Guanine is required is here. Mistakenly, if thymine is coming over here, so this will be recognized and detected by exonucleases, and they will remove this thing. And they will bring correct nucleotide over here, nucleus, and they will bind with it. Suppose this is adenine, and the thymine is required over here. Mistakenly, if guanine is coming over here, so exonucleases will remove this guanine and will bring correct and the required nucleoside over here to bind. So this is called these are the enzymes which are monitoring and carrying the replication in bacterial GMF. In eukaryotes, if you will use the enzyme, 5 common DNA polymerases from mammals, polymerase alpha, nuclear DNA replication, no proofreading, in mammals if you will see. Polymerase beta, nuclear DNA repair, no proofreading. Polymerase gamma, mitochondria DNA replication with proofreading. Polymerase delta, nuclear DNA replication and proofreading. Polymerase epsilon, nuclear DNA repair with proofreading. DNA repair is question, but proofreading is doing. Different polymerases for the nucleus and mitochondrial DNA. Some polymerases proofread, others don't. Some polymerases used for replication, others for repair. So polymerase enzymes are present in bacterial cell, in every cell, either that is eukaryotic cell or bacterial cell. These are controlling the all process of its replication. These are reducing its mistakes or mutation chances. So we are discussing an example in prokaryotic cells. Origin of replication, how it starts replication in bacterial cell. Examples. It begins with double helix denaturing into the single strand, thus exposing the bases. What happened? Because DNA double-stranded helical structure, it it is double helix will be loose and it will convert into the single strand for shorter. Each strand will be separate. So each strand will open and a bubble will form. Begins with double helix denature. Double helix denature will be structure will be denature and it will convert into single strand and each strand will be exposed to the nucleotide bases. Exposes a replication bubble and uh, it will looks like a replication bubble from which replication proceeds in both directions. This is the phenomenon. Suppose this is bacterial DNA. It is its helical structure has been unwinded and converted into single strand. This one is single strand. This one is double strand. And the bond between these two strands, either between adenine, guanine, thymine, cytosine, bonds has been broken down and has been converted into single strand. Now this place is open for to start the synthesis of new strand from here. First new DNA chain made at origin. This one. Replication origin sequence. DNA and this is RNA primer because replication never starts with nucleotides. For this, RNA primer is required. So first a small sequence of RNA will form and this RNA sequence will provide the first hydroxyl group to attach the first nucleotide over here. This RNA primer will provide hydroxyl group to bind the first nucleotide and the first nucleotide when will bind it will provide the place for second, third, fourth, fifth, sixth and this chain will increase. Similarly, suppose 245 base pairs in E. coli, progression to two replication forks moving in opposite directions. So replication takes place once a DNA double helix structure will be unwound and it will provide place for the synthesis of new daughter strand. So it is synthesis with place in two directions. So This is also open. Every open direction will providing a template, and every template will work at the same time. So when replication starts from here, it will go in this direction because this side is also opening. So this will go in this direction. So because this will open more and more, so this strand is also opening. RNA primer again is coming over here, and it is synthesizing uh, daughter strand. It will open gradually and it will synthesize uh, continuously. It is going to be open after some time when there will be sufficient space. RNA primer will come and it will make a small fragment of the DNA. So replication will go in this direction and replication also will go in this direction. Initiation of the replication major elements, if you will see, segments of single strand DNA are called template strand. Segments of single stranded DNA are called template strands. So these are called template strands. This is also template strand. This is also template strand. This is also template. This is also template. Gyrase, a type of topoisomerase, relaxes the supercoiled DNA. So what happens first? Gyrase enzyme is coming. It will bind with DNA, which is in helix form. It is a type of topoisomerase. It will relax. It will unwind the supercoiled DNA. Unwind, and it will generate the single strands like this form. Gyrase will form this form because this will be the first site from where DNA synthesis will take place. Initiator proteins and the DNA helicase binds to the DNA at the replication fork and untwists the DNA using energy derived from ATP, adenosine triphosphate. Hydrolysis of ATP causes a shape change in DNA helicase. So this will initiate. First, when gyrase will come, it will bind with DNA. It will make it loose from one side. It will release or it will relax the supercoiled structure of DNA. When it will be relaxed and it will convert into a small place, it will convert into single strand, if you can understand. So initiator proteins and the DNA helicase binds to the DNA. So some proteins will come and these will stop to rewind it. Initiation proteins and the DNA helicase binds to the DNA at the replication fork and untwist the DNA using energy derived from ATPs. DNA primase next, and what will come? DNA primase next binds to helicase, producing a complex called primosome. Primase is required for synthesis. So primase synthesize a short RNA primer range from 10 to 12 nucleotides to which DNA polymerase 3 adds nucleotide. So here we have to clear that primase first initiation will be started by primase because it will form a small RNA, a small synthesize a short RNA primer, 10 to 12 nucleotides, because it will provide free hydroxyl site for binding of the coming nucleosides, to which DNA polymerase 3 adds nucleotides. Polymerase 3 adds nucleotides in direction 5' to 3' on both strands, beginning at the RNA primer. So it is the limitation of the polymerase 3. It adds nucleotides from 5' direction to 3' on both strands, beginning at the RNA primer. So RNA primer is removed. At last, RNA primer will be removed and replaced with DNA by polymerase 1, and the gap is sealed with DNA ligase. Single-stranded DNA bind SSB proteins stabilize the single-strand template DNA during this process. This process is called elongation. So this is the process, model of the replication in E. coli. Maybe we have to discuss this thing in our second class because it is quite enough for this class. So from here, model of the replication in E. coli we will discuss in next class. Thank you. Have a nice time.